Ready? Okay, yeah, thanks. Good evening, everyone. Welcome to another session on docking. And today we are going to see about AutoDock. I know the topic is about advanced AI docking tools. We'll see some of them as well. Since most of you have Windows-based tools or Windows-based laptops, or some of you may have Linux-based. There might be some of the tools which need a little bit more command or command level or advanced knowledge of Linux commands for setting up an installation and all that. So demo is going to be a little bit more simpler so that everyone can replicate it and everyone can work around with that. So this is a recap of what we saw last week about docking. Last week we saw a little bit about introduction and the way molecular docking, the basics and all. And then I also went through the same slides, but today I'm going to go a little bit more in deep depth about one particular tool which is quite popular and it's very easy to use, and it has a very good um, way of representing the data and has a lot of uh, options for you to try at different levels, whether you are from the basic level or whether you are intermediate or whether you are advanced. Okay. So Autodoc is a method that uses one of the optimization techniques called genetic algorithm, and it was developed by A.J. Olson and his group in 1990. And before that, the methods that were available was doc, and there was FlexX, there was ICM doc. Some of these are commercially available tools. So there were doc was not commercially available. It was academic free. And so Olson wanted to use a much better approach of conformational sampling. And once they had the conformational sampling done, they could use a new scoring method to find out the free energy of the dot molecules using a grid-based potential. Okay, so the main advantage of Autodoc was that they were able to implement three different algorithms for conformational search in the same package. The first one is simulated annealing. Now, simulated annealing is something that most of you would be familiar with, not with the first word, but the second word. So when you say something like you are annealing something, so annealing means that you are allowing bonds to be reformed when they are um, yeah, when bonds to be reformed when they are broken. So the easiest example to understand annealing is by looking at glass or molten glass. Okay. So when you heat glass at very high temperature, it melts, it becomes liquid. And then when you are making a sheet of glass or when you're making a lens, you have to bring the temperature slowly down. You cannot drastically bring the temperature to a very low temperature immediately because that will develop cracks. And the reason you develop cracks is because the bonds between them gets broken. Now, and this is something that you would have experienced in your daily life when you have poured hot water in a glass and then you're immediately bringing the temperature change, the glass breaks. Now, in the case of proteins, simulated annealing, or in the case of ligands, simulated annealing is done where you are also using the temperature as a factor. But when you're trying to bring down the temperature 1 degree at a time, and you are bringing the temperature to a particular limit, like for example, room temperature or the human body temperature or 0 Kelvin, okay, where everything is frozen. So what happens at that point of time is that whatever bonds were broken, they will slowly start making up. And the usual way in which proteins, when you blow up the protein and then when you're trying to anneal the proteins, it will try to reform the bonds that stabilize it. And that is the whole hypothesis of this used in docking. Because when you are fragmenting the molecules or blowing up the molecules, you get different fragments. And then when you are bringing the temperature down, you will see that the most favorable interactions will be the first ones to start making a bond. And they will start forming more and more, and they might start becoming lengthier. So, this idea ends up creating new conformation every time you increase the temperature and then decrease the temperature and so on. And so this was the idea behind implementing the simulated handling. And all these algorithms, excuse me for 1 minute. I have to address something in the next door, but I'll come back 1 second. Sorry about that. So these algorithms are used for, as is written in the slide, for search algorithms. And like we saw last week, molecular docking involves two steps. First is conformational search, and the second step is finding the score of the conformation. So conformational search is an exhaustive process, and we saw this with the Anifsen's experiment, the thought process, the Levinthal's paradox. So we know that you cannot search all the conformations to find the lowest energy conformation. Rather, you want to find an approximate, or you want to find the lowest energy conformation in the shortest possible time. And that the way you can do that is if you know what the energy landscape is. But when you take a new molecule or when you take a new protein and then you have not done free energy calculations for it, you have not mapped the conformational search space, then there is a very high chance that whatever conformation you're going to take may not have the lowest energy for that. And so it is advisable that you take the sampling of the conformation in the valley. We remember the valley, and where in the hills is the highest energy. And as you go down the hill, you are having lower energy, and you have multiple minimas. And so when you are traversing different areas in this valley, in this energy landscape, you want to come across the lowest energy within the shortest possible steps. And one way to do it is simulated handling. And the second way in which you can do the same conformational search is through genetic algorithm. And the third one is a Lamarckian genetic algorithm or local search. Okay. So these are the three things that Olson's group introduced, and it became very popular because now you can select different types of algorithms to do the same process. So again, whatever I described is what is shown in this slide. Simulated handling is based on temperature effects. You start with the high temperature and then you do a global search of the conformations and you do a local search with the lower temperature. So with lower temperature, very minimal torsions will be changing, very minimal rotational bonds will happen. And so you can find out which is the optimal conformation that can bind to that particular target. Now, genetic algorithm is an algorithm that is inspired by biology, and it actually is now used in biology. When in fact it was not made for biological applications. So when computer science they wanted to find out what type of optimization rules can be made or what type of optimization methods can be developed. They took biology as an inspiration and they saw that genotype dictates the phenotype. Similarly, in the Lamarckian view, the phenotype dictates the genotype. Well, we know that Lamarckian theory of evolution is disproved. It doesn't work in many cases, in most cases. Darwin's theory of evolution is what is the best model that fits the evidence that we have. But here, computer science uh, people or the optimization people, they understood that you can define an entity as a gene. A gene can have alleles, and these alleles can be come up with different combinations. And that's what they were trying to see how they can implement that with the optimization problem. So, algorithm model for simulated annealing is like, as I said, explained by glass. So, when you cool a solution of liquid molten glass, it forms a crystal with ordered molecules. If you cool them very fast, you get cracks, and that cracks is because you have a disordered system. What you need is a highly ordered crystal, and this highly ordered crystal, in the case of ligand or a drug, is the molecule's lowest energy state because that is what is comfortable with, and it is where all the bonds are satisfied, all the valencies are satisfied, and it is pretty happy about it. So either you can simulate through a linear slope cooling or a proportional slope cooling, and you can do a lot more things with it. So when as the temperature increases, what happens is the molecules that you can find at very high temperature also increases. That means you can find more solutions with a particular distribution curve. So you generate a set of solutions, new solution is compared to the previous solution, and if the energy is lower than the previous, then you accept it. If it is higher, you discard. But you have a certain um, probability of accepting a higher solution or a solution with a higher energy because you want to traverse the conformational landscape. You don't want to be stuck in a local minimum. You want to go to the global minimum. So if a new solution is high, has a higher energy, you accept it probabilistically according to a Boltzmann distribution. So you don't reject all solutions with a higher energy. Sometimes you do accept them, but with a very low chances. So if this is the cutoff, okay, so for something, the kinetic energy and the probability, you can you can find out what the energy of that particular conformation is at this end of the graph. When they are in this particular side, they are of higher energy or they are of like a different conformation. And it depends on whether you want to check whether the previous step or the next step is much having lower energy than the previous energy. So Boltzmann probability distribution is like, I don't want to go into the details for you, not to confuse you. For your level, if you understand that the Boltzmann probability distribution changes with respect to a given solution, and you have a probability of accepting a solution or not depending on the distribution then. Okay. Now, in the case of genetic algorithm, this is something that I showed you last week also. Imagine you have a set of genes or a population having 6 genes, and each gene has 2 alleles, a binary allele, ones and zeros. So sorry that the alignment of the numbers are not in the boxes. So imagine it's a box, each box having a value 1 or 0. Now, just like how you have a recombination event that you have in a gene where there is a crossover, there is a mutation, there is a selection of a particular mutation or rejection of a particular mutation. You have these kind of operations done at the binary level also. So, for example, you can say that, okay, this particular set of genes, these 6 genes, they give a score of 20. This set of genes, they give a score of 13. Okay. And this set of genes give a score of 48. And this set of genes give a score of 52. Now, you have a fitness function. This fitness function is again a probability distribution. The probability distribution says that you can accept or reject a particular selection. Okay. Now, what do you accept? You accept a score that is higher in nature. So a score of 13 may not be selected, but a score of 20 will be selected. Score of 48 may not be selected, but a score of 52 may be selected. So the operator chooses those individuals with a particular cutoff. Now, in this particular fitness function, you can either flip the question of either selecting those with the higher scores or those with the lower scores. And this slide, it says that it's selecting that those with the lowest values. What happens is that the chance of 52 getting selected may be very less, but the chance of 13 getting selected is pretty high. But what about 48 and 20? Those with higher values will be chosen with a very low probability. So 48 and 20 can still be selected, but not as 52 may not be selected that frequently. Given this, when you have a molecule, when you have the bonds and the torsions and other parameters which are declared as genes, this is what I told you last week, you can change those parameters and you can recalculate the score. And when you recalculate the score, you will be able to find out whether that score is higher or lower than the parent. So you got a new score, which is your offspring. You will select based on the previous, the parent. Now, in evolution, in biology, what happens? The parent transfers the good genes or the genes which have a higher fitness function to the offspring for them to survive and for them to propagate the benefits of the evolutionary advantage. So in that particular case, the offspring should obviously have a higher fitness function than the parent. Then only it gets accepted. You can see this in animals. In biology, what happens? You let us say, I don't know how many of you have pets, and if you have pets, I don't know how many of you have seen the behavior of birds. They will lay many eggs. There will be among all the eggs, there will be few that are very healthy birds, few that are not very healthy, they are weak. So the fitness there, the evolutionary fitness is the energy spent on feeding the weak bird is not advantageous for the parent. So what the parent bird does is that it slowly pushes the weakest bird, or it knows that it won't survive the winter or it won't survive the climate change, climatic seasons. So it will push it out of the nest. Among all the ones that are surviving, even among that, you will see that one will be a dominant chick, the others will not be a dominant chick. They will be eating more food from the mother bird than the other siblings. So that fitness function enables that the weak progeny is not is out of the system, so that if there is, let us say, not an evolutionary advantage, it will not be propagated to the next generation. So this is again used in this genetic algorithm optimization method, where a conformation of a ligand which has a much, let us say, has a higher energy than the parent, it will not be accepted. If it has a lower energy, because lower the energy, the better. You want to find the conformation with the lowest energy or the minimum energy. Then you can select that particular conformation. So what you select, how you select, and what probability with which you select a particular offspring in the genetic algorithm are the parameters, and that is fine-tuned, that can be changed based on system to system. Okay. So the progeny in the case of genetic algorithm or an offspring is that you can switch the ones and the zeros, like in this image, either a huge stretch or only one particular stretch. When you change a huge stretch in or a huge number of genes, you change a crossover. What happens in the energy landscape? You are here and suddenly you jump to another place. If you change only one parameter or one gene at a time, imagine, keep remembering that genes are nothing but bonds, angles, torsion angles, and all the things that describe the molecule. So let us say you changed only one thing, like a rotational bond. So in the energy landscape, you may not be able to jump a huge jump from one place to another, but you may be making a small jump from one to another. Okay. So that kind of change is also acceptable, depends again on if the offspring from the parent alleles has a benefit in terms of lower energy. I'm pretty sure by this time you understand how it works. Okay. So I don't have to teach a lot of biological concepts to you to make you understand genetic algorithm. But the beauty is that something that we observed in biology was developed as a computational algorithm to find solutions among a set of solutions. Okay, so that is the optimization problem. Every solution is a good solution, but you only have one optimal solution. So you want to find the optimal solution at the lowest, shortest time possible, like the traveling salesman problem that I told you last time. So this particular genetic algorithm, it so happened that Olson's group found out that they could use this for a biological problem. So it's like a circle come back, a biological phenomenon got written as a algorithm, and that algorithm is now used in biological applications. Okay, so that's what I want you to understand. And there are different types of technical terms that you see here. Like for example, uh, you the offspring at one after one cycle you do multiple cycles, and after one cycle you replace the parental generation. It's basically the parental generation are dying off, and you have the new offspring which are now the parents, and they give more offspring. Now elitism is a step or a parameter in setup in genetic algorithm where the best individual from the parent generation will persist if it is a better solution than the new individuals created. So what happens is the longevity of that particular parent, because they had a better score, will be continued to the next generation. So they are living for a longer period of time. Okay. And this process is repeated until you find an offspring or an individual that has a very good fitness function and a very good score. So in Autodog, as I said, the genes are its X, Y, and Z coordinates. You can have a rotational bond. The rotation is usually 360 degrees, but because of the constraints in the ligand, some of these are not 360 degrees, but maybe 90-degree movement, or it could be within 180-degree movement. Whatever the rotation it is, you have the XYZ coordinates as genes. So that means every time you are changing an X coordinate, keeping Y and Z constant, which means that you are moving the molecule on the energy landscape on the Y axis. Sorry, on the X axis. Similarly, if you change all three, that means you are going to jump from one part of the energy landscape to another part of the energy landscape. So when you do this, you are actually looking at a lot more conformational space, and you will be able to do this even if you're stuck in a local minima. And that is a big problem in biology, especially for simulations. And the only way we can come out of the local minima is when you give energy to it. Like, for example, an enzyme catalysis. You heat up the enzyme, it catalyzes faster because it can go over the hill, the barrier. Same concept, but in the conformational search space, you are changing the XYZ coordinate completely so that from the local minima you can escape and you can jump to another place in the energy landscape. So this is the energy landscape. It's sliced in a two-dimensional plane, but What I want you to imagine a conformational search space with energy is energy is in the y-axis, conformation is on the x-axis, and then you have high energy here, lower energy here, and then much lowest energy here, and so on. So when a parent is undergoing a mutation and a child is getting created, then you are basically jumping the energy landscape from here to here. Okay, so the child has a much better fitness function than the parent. So according to the figure here, the Lamarckian genetic algorithm finds the lowest fitness function values first and then maps these values to the genotype, just like how Lamarckian theory of evolution works. We don't go from the genotype to phenotype, but phenotype to genotype. So you get a much better performance than the simulated handling or just the local search genetic algorithm alone. Okay, so in in the case of Autodoc, you have the first step after you process the file and you have the files ready in the format that is required, which we will cover in the demo shortly. You need to have a create a grid, and the grid is something that is uh, three-dimensional in shape. It has a specific number of points in the x, y, and z coordinate, and each length of the grid has multiple grid points, and each of these grid points are separated across in an ordered manner with a specific distance. So what happens is whatever you define within that grid is what is going to be considered for docking. And when you create this grid, you can specify it as small as possible or as large as possible. And when you specify this as large as possible and the entire protein is within this grid, the three-dimensional box, then we call that type of docking as blind docking. Why? Because you're not focusing on one part of the protein or the receptor or the drug target where the molecule has to bind. You are letting wherever it wants to bind, it can bind. Then you will ask, how do we know if that particular uh, place it binds is a good place or whether that is a place it is preferred? Now, the basic hypothesis is that if you have a good binder and if you are doing a blind docking, the good binder will find the place where it needs to bind with a very high probability or a very high chance. And you would be able to find the place that it binds to with some statistical measure. So I'll again, as I said, I'll cover all this in the demo. But in general, we know in a protein like enzymes, the active site where the catalytic function happens, where it makes an interaction. So sometimes you can have the grid only for that particular region. And that way, your space where the molecule can go and bind and change conformation and do all manipulations, it's very small in area. But when you give the entire protein, it has more freedom to do more number of conformations. So usually blind docking is done when you are not sure where the molecule will go and bind in the protein, but you are interested to know if there are alternate sites on the protein in the receptor in the drug target where something else can bind. So, or if you let us say you don't want to have a bias towards a particular place in the molecule where you want the molecule to bind, because you don't know how they bind in reality until unless you do X-ray crystallography or you solve the structure through one of the experimental methods, drug protein complex, you have no idea where the molecule is going to bind. And even if you do a validation, let us say with molecular dynamics simulation, you will not be able to tell them with good significance or good confidence that this is where it is going to bind. So the grid preparation, you need to think about a lot of factors when you're trying to do the docking. Is this the active site? Is this functional in nature? Does this site have any flexibility in it? If it is has any flexibility, like how much of the flexibility is incorporated in the docking protocol, and what type of molecule it likes to bind to, and is there a crucial residue it is making an interaction with, and so on and so on. And all these questions are not standard, meaning that for every system, let us say today I'm trying to do a molecular docking simulation with lysozyme. Tomorrow I do it with a capsid protein. The strategy, the list of reasons for selecting a particular site as for me to make the grid will change from system to system or from case to case. So, in simulated annealing, you are looking at this type of parameters. There usually you get two types of files, a GPF file, which is made for the first step where you prepare the grid files and you run AutoGrid. And once you have done AutoGrid, you get the map file. So wherever the molecules, the protein or drug targets atoms are in line with the grid point, or whatever atoms are there within the grid point, they are all mapped, and you get a huge file that has a list of all the atoms and the surface area, the electrostatistics, the charge distribution, all that is given in that map file. So you basically generate multiple map files. Ideally, you will generate for whatever atoms are there in the drug target, you will generate a map for that. So you will have a carbon map, you will have a hydrogen map, you will have an oxygen map, and so on. You can make the map for the ligand also. So from the point of the ligand, what should be the receptors map? That also can be mapped, and you will get multiple files. Then you have this DPF file or doc parameter file in which you will have these settings set. Now, this is a flat file, meaning that this is something that can be opened with a notepad or with a text editor, and you can actually edit them even if you did not see a setting where it was supposed to be changed. You can do that. And this is pretty customizable, so it becomes very easy. So this is something that I showed you last time. And this is the one structure where they tried multiple different parameters, and they found different solutions for when they use different algorithms. Okay. So again, the docking parameter file will have genetic algorithm, will have a different set of parameters. The local search will have different set of parameters. Um, since you guys are starting, coming to know about this, at this particular point of time, you don't have to worry about what these parameters or what is this number that I should put there, because we are going to use a default setting and a default value. It's perfectly fine for now. Now, let us say you find for a particular default value, the docking is not proper, or it did not give you the result that was expected. Then, or it did not bind to the particular site where it was supposed to bind because it did bind in an active site. So what do you do next? Either you can go for validation setups and then see whether that is really the reality, or you could actually uh, change the parameters in the docking parameter file and rerun it and see whether you are getting a convergence. Convergence means that you are getting the same result from different methods. So when you have convergence, you get a confidence. And when you repeat this multiple times and you get a convergence, then that is much closer to reality than the arbitrary default values that you had used earlier. Okay. So with this, I will stop the presentation and I'm going to switch to another laptop to take the demo. So at this point of time, I would welcome uh, any all of you to like ask questions. There's about like 10 minutes time before I go to the next one. So go ahead, unmute yourself or put your message in the chat box and ask me if you have any queries or questions. Any questions? Any doubts?
Hi, sir. Actually, I do not understand grid box. Like, what is its like function? Only for locating specific proteins to a particular grid for search.
Locating specific areas in
Sir,
the for
does
doc.
grid means X, Y, Z axis?
Grid box is
Grid.
three-dimensional. So XYZ.
Oh,
Yes. Grid box, the box is
oh.
three-dimensional, and I'll show you how the box looks like. But basically, you will have um, you are looking at a specific place in the protein where you want to do the docking. So how do you define that place? So the grid box helps to identify that particular place where you would do the docking. Any other question?
Sir,
Yes.
sir, does this grid system takes into account the for auto docking physiological conditions because the aquatic environment is always present with certain ion concentrations?
Right. That's a good question. So when you're doing the docking, the solvent is never part of the system. Okay. As we saw last week, you the let us say a protein has a cavity. Okay, so this is the cavity.
Creeks and crevices and
Yeah,
tunnels.
yeah, like canyons. Yeah.
Hmm.
So now when nothing is bound to it, you have water molecules attached to that. Okay.
Hmm.
You have to displace those water molecules and then make something bind to it. So basically,
Okay.
until unless you know for sure that to undergo a particular reaction or to do a particular reaction, water molecule acts as a nucleophile and it donates electrons. You usually do
Oh
not
yeah.
keep water molecule in the active site while doing docking. Okay. So this
Hmm.
goes for other proteins. Like for example, let us say this is a metal ion binding protein. Okay. And
Hmm.
it requires zinc for its catalytic activity. So without
Hmm.
zinc, it is not going to be functional.
Hmm.
Then you should have zinc in the active site, and it is at that place you want to do the docking because in reality, what happens? Reality, the protein will be bound with zinc, and whatever binds to it will also make probably an interaction with zinc.
Hmm.
So zinc has to be present. So we need that
Okay.
kind of empirical evidence to keep certain ions or water molecules or metals
Hmm.
along with the receptor. Now, let us say it is a structural protein. A structural protein means it's not going to do any enzymatic catalysis. It is just for structural support, gives stability to the entity. But the
Hmm.
structural protein is a drug target. Okay,
Hmm.
most like viral capsid protein. Capsid protein, what is the function? It keeps the whatever is inside the cap
Shell.
in the shell intact.
Hmm.
That's the function of it.
Hmm.
So capsid protein doesn't do any enzymatic catalysis. So in that case, if that is the drug target, then the place where you're making the molecule go and bind will not need a metal ion, will not have water molecule. You don't need those kind of things in that particular area where you're going to do the docking.
Hmm.
So you don't need
Yes,
to
sir.
think. So again, the requirement for a particular setup to have certain molecules present in them should have a experimental evidence to it. So most of the time, the general rule of thumb or the general practice is to remove all water molecules, all ions, all crystallography related
Mm.
artifacts, and then just keep the protein alone. And it is through that you do the dock. Now, you asked about physiological condition. Now, let us say this particular protein is active function at low pH.
Say
Then
in stomach, where acidic pH is.
acidic pH. Okay, so let's say at a low pH, acidic pH. Now, proteins, depending on the pH, certain amino acids can get protonated. Yes.
Mm-hmm.
And
Yes, sir.
which amino acids can get protonated? You can look at the pKa curves and you can find
PK
out,
values.
yeah, pK values, and you can find out which amino acids are protonated. Now, when you are setting up the receptor, if those residues are going to that are protonated are going to play a role in the binding of the drug, then there is a reason for you to
Mm-hmm.
protonate it. Now, let us say this is my hand is the protein, this is the cavity, this is where the
Mm.
molecule is going to bind, and there is
Yes,
something
sir.
else on this side that's getting protonated. I really
Mm.
don't need to protonate this residue here because molecule is making an interaction in this side of the protein, not in this side. So that
Mm.
grid, what you select, it is inside this, all these considerations have to be done. So if
Mm.
something is not required, you don't need to do it because it will be an additional setup or you are going to add an additional computational complexity to it. But if you have an experimental evidence and empirical evidence saying that this is required, this should be there, then it's best to have that incorporated and see with and without it. So, because again, with there might be new bonds may not form. Without it, new bonds may be formed. So then which one do you rely? Of course, you rely the one with the metal ion or with that particular experimental evidence because that is more closer to reality. So when we do molecular docking for a drug target, I give this analogy in every class that drug designing is very similar to plotting an assassination attempt. Like for example, how do terrorists plan assassination of any high profile person? The first thing that they do is they go on a reconnaissance. They look at the person's schedule. They see which particular place or which particular day, which particular time of the day they are alone or they are vulnerable, and that's the time they
Mm.
attack. Yes.
Yes, sir.
In a drug target, this drug target is a host protein that is doing its function. Which area in the protein is most amenable for the inhibitor to bind? That
Mm.
is going to be preventing the host protein to do its function. So, the more you know about the host protein, the more clarity you will get as to how to do the docking. And again, remember in molecular docking, we don't ask the question where you want to do the docking. You ask the question how it is going to dock, what type of interactions is going to make, what is the energy of the lowest binding conformation. So those are the questions you're
Mm.
asking. But now we know with many different methods and many different setups that sometimes blind docking helps. And I'll show you how to do blind docking also. So when you do blind docking, you are removing the bias that this is where exactly I want the molecule to bind. That may not be the same that's happening in reality. क्योंकि जब आप एक प्रोटीन ले रहे हो उसको सॉल्यूशन
Mm.
में डालना है और वो केमिकल आपने इनिबिटर को एक पर्टिकुलर टाइट्रेशन में आपने उसको सोक किया है
Mm.
तो वो मॉलिक्यूल कहीं पे भी बाइंड कर सकता है. इट्स नॉट नेसेसरी दैट इट विल बाइंड ओनली इन द एक्टिव साइट. अगर उसको एक्टिव साइट में ही बाइंड करना है तो वहां पे भी बाइंड करेगा, लेकिन कहाँ बाइंड करेगा इसका कोई प्रिडिक्शन आप नहीं कर सकते हैं जब आपको पता ही नहीं कि वो प्रोटीन में कहाँ कहाँ कैविटीज हैं. सो
Mm.
कुछ स्ट्रक्चरल प्रोटीन्स ऐसे हैं जिसमें आपको नॉन फंक्शनल साइट्स में बी द ड्रग टारगेट एरिया एंड फंक्शनल साइट्स may not be the target area क्योंकि the structural protein binds to another protein and they form dimer-dimer interactions, and that site is not available in reality at all. और आपने वहां पे डॉकिंग पूरा किया और फिर बाद में पता चला कि वो अच्छा वो रीजन खाली था ही नहीं, क्योंकि वहां पे वो खुद अपने दूसरे कॉपी से वहां बाइंड कर रहा है, तो वहां पे वो जगह है ही नहीं। सो
हम्म
ऐसे बहुत सारे केसेस हैं जिसमें यू हैव टू नो इन डेप्थ इंफॉर्मेशन अबाउट द ड्रग टारगेट, एंड कभी कभी उसमें पता भी नहीं चलता है कि कितना इंफॉर्मेशन हमें पता है, क्योंकि you don't maybe like read all the literature. Some literature may be in not accessible, or the results are written in a way that you think it is something else. So in those cases, blind docking helps because you are removing the bias and you are saying, okay, let me see which sites on the protein are amenable for docking, and then let me see whether they
mm.
are one among them is statistically significant, and whether you can come up with a strategy or a rationale to identify that particular place as a among the best among all the available options.
Mm.
So that is why this rational drug design is like you can you have to come up with a strategy. You understand? So that
Yes, sir.
strategy needs to be formalized. It has to be really good. So there is two questions in the chat box. Orthodox screen.
Thank you, sir. Thank you.
Thank you, thank you. Orthodox screen the entire protein blindly for active site and show the hotspots for binding of ligand. Uh, the sentence is I didn't understand the question, but I understood the intention. Are you saying that? Are you asking if if by blind talking you can find out where the active site is? Is this what you're asking?
Like it will screen the entire protein to search where is the active sites, like how many active sites are present and what is the efficient active site for binding.
Yeah, that's what blind docking we usually end up doing. If we have no information about the drug target and it's like which particular place we want it to bind. And you had asked another question as discussed earlier, if the protein spaces has lowest energy, it has highest binding capacity. What do you mean by protein spaces?
The regions where the lowest energy is there, so that spaces have the active, like highest efficient to bind the ligands, right?
No, protein, see, okay, protein is already in a lowest energy conformation.
Okay.
Okay. For example, I can function very well if I'm comfortably sitting on a chair. If I'm doing acrobat and मैं शीर्षासना करके मैं काम नहीं कर सकता. Okay, I'm not in a high energy. I'm in a high energy state. I'm not very comfortable. I cannot do any work there if I'm मतलब उल्टा लटक रहा हूँ. So the protein is already in a lowest energy conformation, which is functional. For a protein to be functional, it has to be in a lowest energy state. This is a fact. Now, when we say these words, we don't mean that the surface of the protein has low energy or pockets of energy. Okay. The entire conformation, when you calculate the total potential energy or the total kinetic energy, that will be lowest in value. That is the entire sum of the entire protein. What we mean by low energy conformation is that either for the protein and for the ligand, both have to be in low energy for them to bind optimally. Okay, that is the low energy conformation that we are talking about.
Okay, so autodock helps in both docking of ligand to a protein and a protein-protein docking.
So again, remember, go back to the previous set of slides, not the one I showed today, but in the previous week, where you do rigid docking and you have rigid flexible docking. So we usually do rigid body docking when both are proteins because both are big molecules. But docking, usually you do one which is relatively larger, one is relatively smaller compared to each other. So you dock the smallest one to the bigger molecule. Now, when we dock the smallest one to the bigger molecule, the smallest one, or the one that has small size, you can introduce flexibility in it by looking at the conformational landscape for that particular molecule. And when you do that, you will be able to find the low energy conformation of this molecule that goes and binds. Now, this molecule is rigid. So you keep this rigid, the big molecule, you make the small molecule flexible, and then you make the docking. Now, you may ask if this is in low energy conformation. Yes, that is assumed that the conformation that of the host target or the drug target is already at its lowest energy state, and it is ready for something to go and bind to it. And that is why this, even if it's this in high energy, it can still go, we can find the low energy for this and go and do the docking for that. So we cannot, if we want to find low energy for both protein and ligand with the starting conformation where both are at a high energy state, then the computational intensity is very high. You have to simulate both flexibility and you have to look at two energy landscapes at the same time. And these are pretty time intensive tasks. You can't finish it on time. It will take a very long time. Now, if it is like only screening one molecule or looking at two molecules, then yeah, okay, it's good enough to spend that much time on the finding the conformation for both. But if you have 1 million compounds to screen and you want to find which is the good binder, and you want to shortlist maybe 100 molecules among the 1 million, which you can synthesize in the lab, then the time taken to find the flexible docking is very, very high. And no one does that because in industry, the motto is fail fast and fail quick. So if you have multiple options, you want to see which options, you will not see whether option one will succeed. You will see how quick option one will fail. If it fails fast in the first step, you eliminate it. You go to the next step, next option. When that fails, you go to the third option. I like that. So it saves time, it saves money, it saves energy. Because think about industry when they are like, for example, I don't know how many of you have come across this in the recent past that Wockhardt industry is a big pharmaceutical company, had pumped about 150 crores and came up with a new antibiotic, which is probably going to be launched commercially only like 2 years from now. But they are very close to getting the FDA approval and they have got the FDA approval for that. And but the amount of money they had spent on that one particular antibiotic was 150 crores. But they did not start with one antibiotic. They started with millions of possibilities. And then they came up with one drug molecule, which was like was good on all criteria. They, a similar molecule which had a different functional group ended up being hepatotoxic, toxic to the liver. So they had to reject that candidate. But all this information you can find. But the reason I'm saying is, let us say you are working on this type of project and you want to find which molecule can cause liver toxicity. If you find in the very first day or within the first couple of minutes that this molecule is going to be liver toxic or hepatotoxic, you don't want to continue down the path of asking the question how to prevent the hepatotoxicity. Rather, you would ask the question among the 10 molecules, which one causes less hepatotoxicity compared to the other 9? So that ends up saving a lot of time, a lot of money, a lot of manpower, a lot of energy, and more such questions are asked, you will be end up saving time in the entire drug development process, and you will be able to like release the product much quicker. So that's the whole idea of removing or eliminating bad binders as quickly as possible and keeping only the good binders. So in virtual screening, we do enrichment where we see whether the good binders were really enriched. We find really good binders among the best, among the decent enough binders, and we have eliminated all the bad binders. I think we are close to the time for me to start the demo. Let me start it. Okay. Okay, so you should be able to see a screen, another screen, not the one that I'm talking from, but another screen. And what you see here is the Autodoc tools. This is the GUI for Autodoc, and this works in Windows and Linux. This doesn't work in Mac, but this was developed by a completely independent team that actually wanted to make things easier for people who run Autodoc and set up the entire process, like people who are new to it. Okay. So at the top, you have a menu, file 3D graphics, and that is the menu for the MGL tools. That is the interface. And the Autodoc itself has its own menu, the one in brown, ligand, flexible residue, grid, docking, run, analysis, and so on. So the first thing that I need to do is set the settings. So here I have a directory called docking under the A drive. So I'm going to say copy this path, and I'm going to put it here, and I'm going to make default. Why am I doing this? Because by default, this will be pointing to program files or the place where the executable is present in your C drive. But that is not where my input files are going to be. My input files are not going to be in program files, Scripps Research Institute, Autodoc. Enter the Autodoc folder. It is going to be in some other folder in my documents or in my desktop. Now, that is the one reason where I have to make this as default and set this path so that when I open a file or when I go and say open a new file, it will automatically look into this directory where the files are supposed to be, not in the installation path where there is nothing. I don't have any of my input files there. So once I do this, and there's one more thing I forgot to tell you, that the path that you have set for running the Autodoc or whatever docking you want to do through Autodoc tools, that path should not have any spaces. Okay. So if you have any space, then it will not be able to run the tool. Why? Because in Windows, spaces are accepted. Okay, so if you can make a directory, a folder name saying test files. So test space files. You can have it like that. It will still work. But in the Linux system, you cannot, by convention and by a basic rule, is that a file name, a folder name with space means that space is also a character. And most of the time, it is very difficult to put a backward slash and a space and declare that special character. So it's avoided most of the time. And usually, you do not make a folder with spaces. So if you have no space, that's good. If you have spaces, then you have to remove the spaces, rename the folder names, rename the path, and you have rename the path means not just typing and removing it here. You have to remove it at this particular path that is shown here. You cannot have a space based path. So it's a good practice not to have any spaces in the file name and the folder name. Okay. So now in this particular directory, I have two files. One is a PDB file, the other one is a PDBQT file. Now, and this file is where I have all the output files that I've used as examples to show, but I'm going to show you uh, right from the scratch what this is. Okay, so 396 So in PyMol, I'm going to show you what. So this is the protein. Okay, and as you can see, this particular file contains only the atom records. It doesn't have any of the heteroatom records. And you can verify that by looking at the sequence tab, and you will see that there are nothing other than amino acids in this. So this is a processed PDB file from the PDB. So even if you get a PDB file with water, ions, you have to remove all of them, because right now for this particular molecule, this drug target, I don't know if water is needed or any metal ions are needed. I don't know if it needs an acidic environment to work. I'd have very less idea about it. So because I don't know anything about it, I'm going to remove all the unnecessary things except the protein part. Okay. Now, in the same directory, I have another file, which is a PDBQT file of a peptide. Okay, so this is a peptide. So this peptide is like a big, long uh, conformation. There are a lot of disordered regions. There are a lot of ordered regions. Um, Once again, I'll have to mute myself for this. Once again. Okay, so for this particular demo, I'm going to take a peptide. This is an antimicrobial peptide. And as you know, antimicrobial peptides are the latest things that are being promoted for overcoming antibiotic resistance in escape pathogens. Escape is nothing but an acronym that stands for ESKAPEE, and E stands for Escherichia coli. S stands for
Staphylococcus aureus.
thank you, Staphylococcus aureus. K for Klebsiella pneumoniae, and Acinetobacter baumannii, and then Enterococcus and Pseudomonas for the P. Aeruginosa. So all these pathogens, escape pathogens, are pretty notorious in having multi-drug resistance and extreme drug resistance, and there are no known drugs that can act across all the pathogens at the same time. So there are a lot of studies being done. There are a lot of developments happening, and antimicrobial peptides are one of the things that's being promoted. But we don't have, except for one or two antimicrobial peptides, we don't have many that are FDA approved. So a lot of them degrade pretty quick. They're not stable. And so let us say for this particular, I'm taking the DBAMP, which is the database where this ID 10923, and these are the 13 residue antimicrobial peptide, which I have created the three-dimensional structure using AlphaFold. Okay. Now, again, the ligand here is a small entity compared to the protein. So how small it is. So if we have the protein, So this is the protein, and that's the peptide that is we are trying to bind. Now you may see that they are coming close to each other, and you may think that is docking. No, they are not. It is, we are just brought it in one origin space, so it's an artifact, and we don't know how these two will interact. So let us see. Okay. So using these two examples, let me copy this file to the previous directory. Okay. Now this particular file, I showed you the other day, the PDBQT file, the format. So PDBQT is the same as PDB file, PDB format, but with two changes. One is the charge, and that's the Q that stands for. Q stands for charge. And T, it stands for torsions. So what we add here is you have the X, Y, and Z coordinate here. And then you have the occupancy and then the B factor. So at the end, you have a space where you can add the partial charges. So all these are partial charges for these atoms. And in the remark section, you have between which two atoms you have a torsion angle. So you have 27 torsion angles, meaning 27 rotatable bonds. And what is the branching of those torsion angles or the rotational bonds are being shown at the end. So here you see the degree of freedom. Torsional angle, degree of freedom is 55 for the 27 active torsions. You can see 27 active torsions. This was something I pointed out last time, that for Autodoc, the number of torsions has to be kept 32 or less. So this is perfect. It has 27 active torsions, which means it makes the entire molecule flexible. Okay, good, works for our purpose. Okay. So now the first thing that I need to do is I need to prepare the receptor or the drug target also in a PDBQT format. So I need to open the macromolecule. As you can see, it directly opened the docking directory, and it looks for a PDBQT file. I don't have one. So I need to change to all files and then select the structure, and it will start doing some processing by automated way. It thinks that everything is kosher, and it does, oh, it had found these many non-bonded atoms, it merged the non-polar hydrogens, and it gives me a message. I say, okay, fine. And then it will say, oh, now you save this as a PDBQT file. I shouldn't save this as a PDBQT file because I didn't add two things to this. And it will throw me a warning when I say cancel. It will say auto grid requires a written PDBQT molecule. I agree. So what do we do now? This molecule or this protein, I go to the MGL tool tab and I first add charges. Now there are two types of charges that can be added. One is Coleman charges and Gastiger charges. Coleman charges are the ones that you want to add to the receptor. The Gastiger charges are the ones that you add to the small molecule or the ligand. But in this particular case, the antimicrobial peptide is also a protein, right? A small protein, 13 amino acids. So we'll add Coleman charges to both, which has already been done for the AMP. So in this case, I'll add, and it will tell me the Coleman charge added is 10. That means that that protein has a charge of 10. And then I add the hydrogens. For now, by default, I add all hydrogens, and I let the settings or the default settings to remove some of the non-polar hydrogens with it. Or I can add the polar only hydrogens and save it. So it's always okay to add all hydrogens. You want all the valencies to be satisfied. Now we have added the partial charges and the hydrogens. And now I go to grid. Earlier I had selected open, but I already have the object in the virtual space here. So I'll go and choose the macromolecule rather than opening it again, and I'll select it, and it will throw me the same message. And then now I can save it as a PDB-QT file. Now the macromolecule has been processed, and now the macromolecule can be set up for the auto grid, the first step in Autodoc, where first I selected the macromolecule in the same menu. Now I'm going to set map types. Now there are different ways you can set map types. As I said, you can set it directly, or you can select it based on a ligand, or you can select it based on some other set of ligands and so on. And it is in this particular place you can select which of the residues in the receptor you want to make flexible. Now, you cannot make all the residues in the receptor flexible. You can only make certain residues flexible, and that has a limit as well. So you can select up to 10 residues or 15 residues, and that's all that you can do. So right now, since I don't know anything about this molecule or this receptor, I'm going to do a blind docking. So when I do a blind docking, I'm going to let it select directly, and you can see the type of atoms that will be generated, for which the maps will be generated. I accept it, and then I go to the grid box. Now suddenly when I open this particular one, you can see a box here. I'm rotating the molecule and you can see that there's a box. Now the center of this particular box is centered at the macromolecule by default. But let us say you know the active site, which is somewhere here, this particular tyrosine, okay, or this phenylalanine here. Then you can center it on a particular atom. So for that, you can change the XYZ of in the grid box center here, and you can move the box from here to here. Now, by default, you have 40 points in the X, 40 points in Y, 40 points in the Z direction, and each point between 2 points, the distance is 0.375 angstrom. So can someone tell me what is the length of the box in the X-axis? Hmm? How will you calculate? So there are 40 points on X-axis. Distance between each point is 0.375 angstrom. So how many angstrom is the X-axis? Anyone? You basically have to multiply 40 into 0.375. What will be 40 into 0.375? 15. Yes. So 15 angstrom. So the x-axis is 15 angstroms, y-axis is 15 angstroms, and z-axis is 15 angstroms. And that's given by the RGB color. Okay. Now, as I said, by default, it's 40, which means this is the box that the system has given you. But you can increase it up to 126 points in each direction by clicking and dragging it's like a dial, rotatable dial. Now you see the box is amazingly big. So it's 126 points. So what will be the length of the x-axis? 126 into 0.375. How many angstroms? Anyone? You can open a calculator and you can tell me 126 into 0.375. 47.25. Yes. So from a 15 angstrom to 47.25, on the screen it looks pretty big, and it is like 3 times bigger. So now you can see that the protein is 90% inside this box. That means it is this box that in which we will going to set all the maps. Okay. So there's a lot of empty spaces and a lot of places where the molecule can probably go and bind. So let's keep it like this. So I go to File, Close Saving Current. Now, once I do that, the grid vanishes, but actually the information is saved. And then I go and I write the GPF file. GPF stands for grid parameter file. And I usually give the molecule name itself as the grid parameter file. And I save it. Okay. Now, what will be in the grid parameter file? It's a flat file. So you can open it and you can look into it using a text editor. Whatever information you gave, like 126 points on each axis, the spacing, and the type of maps you want to create, what all the maps it will create, the file, solvation map, electrostatic map, all this information are given here. That's what it is. Now, we are ready for running the auto grid. So you go to run, you run auto grid. Now it will look for this particular executable. This is the executable that will run autogrid. So that will be present not in your folder, but in the installation folder. So you have to go to your PC, to the C drive, and then the Scripps Research Institute, Autodoc, and then you have this executable application, AutoGrid 4. So when you select AutoGrid 4, என்ன சீக்கிரமா கிளம்பிட்டு இருக்கேன் அதெல்லாம் எனக்கு தெரியாது என்ன பண்ணிருக்கீங்க ம் எல்லாம் பண்ணணும் என்ன பண்ணணும்னு வச்சுருக்கேன் ஏதோ ஒன்று பண்ணிட்டு இப்போ எழுது அப்படி இப்படின்னு சொல்லி ம் இப்போ நிஷா என்ன பண்ணா அப்டேட் பெற்றல அப்போ நீங்க சாப்பிட்டுட்டு முடிச்சே இல்லையா முடி முடி எதுக்கு கிளம்புற சீக்கிரமா டாக்ஸி பிடிச்சிட்டு போ பஸ்ஸில் தான் போகணும்னு எழுதியிருப்பேன் இல்லை வீட்டுக்கு போயிட்டு முடிச்சுட்டு நீங்கள் ஈவினிங் அனுப்புவியா அதுவும் பண்ண மாட்டேன் பண்ண மாட்டேன் வே பொறுத்து போகணும் மண்ணாங்கட்டி நீ சொன்ன டைமுக்கு நீ அனுப்பிச்சியா okay so you select the gpf file and then automatically it will give the output file name glg. Gpf stands for grid parameter file and glg is a grid log file. So in this file is what the output will be written, and all these outputs are flat files. So now this is the command, what you see here, autogrid4.exe, and then -p is the input file, the GPF, and -l is the GLG. So this is the command that will be running in the background or the backend. So when I click launch, you will see this white box with something running. Okay. Now you don't have to twiddle your thumbs to see what is being done. You can automatically see in your drive, in the folder, that all these map files are created. And this is the output file. So we can open this file and then you will see that there are a lot of things being written, or what are the type of atoms at this particular XYZ axis, it was what type of atom it was assigned, and so on. And all the map related informations will be given, energy terms will be given, and at the end, it will tell you how much of percent was over and what is the time it took. So right now it shows 4%. If I refresh this file, it says 7.9%. So if this grid box was 15 angstrom cube, it would have got over by like like maybe 2-3 minutes. But since we now have a 47 or 42, 42, what's the size you guys told me about? 47.25 cube. So the size is much bigger. So it takes a little bit more time to map all the different atoms that are there in that particular grid box. Okay. So while this is running, this is the first step, or like maybe the second step in Autodoc, where we are preparing the grid and making, we have processed the files, the input files to do the doc. Okay. The next step is the easiest one, which is the actual docking part. Before we go there, and we cannot proceed to that until this finishes, and this has to show 100%. If you have any questions up to this point, ask me.
Good evening, sir.
Yeah.
So is it mandatory to take all the axes in the equal distance?
No,
Like,
you don't need a
or
cube. You don't need a cube. You can be a
yes, sir.
cuboid. You can be a cuboid. Not all
Okay.
drug targets are spherical and globular in nature. The drug tar- the drug target that I used in my PhD was a linear protein, and we had made, like, you if I had to do a blind docking for that, then I have to have a different shape. It doesn't have to be a cube.
Like in this protein, the macromolecule, we have a protein part that is left out of the grid box, right, sir? So can we increase that only
Yeah,
that axis and can incorporate whole protein and can do a blind docking? Maybe the active sites can be reside in that part as well.
you can increase that by increasing the grid spacing. So right now the grid spacing is 0.375 angstroms, right? Between two grid points, the spacing is 0.375 angstroms. And maximum number of grid points you can have in any axis is 126. You increase that 0.375 to zero point 0.5. Then what will happen? Your 126 points will be more spread out.
Yes, sir.
So you can increase the grid box like that.
Does it have any effect, sir? Like any other docking?
The only thing is your grid maps will be different. The earlier 0.375 is a default setting. You can reduce that also so that you'll get more denser maps. When you have maps where the grid points are distantly placed, then the atoms that are between them may not be mapped. The atoms that the grid points are touching, only those will be mapped.
Okay, sir. Okay, sir. Like I have I have this docking class in my semester where my teacher told me like we have to note the grid points as a config file. What is the importance of that config file, sir? Configuration file.
I just showed you this GPF file, right?
Yes, sir.
That is the config file. So where you have yeah, right. So where you have the receptor file, you have the spacing, you have the number of points, and what all types of maps will be created. This is the one that he was talking about.
Okay, sir.
Importance is, this is the input file. This is telling the AutoGrid what to do. What all files need to be created, what are the input that needs to be taken into consideration for setting up docking. You change anything in this, your docking results will change. So this is not just for viewing. If you have a grid parameter file like this, you can change the grid center by deleting, and you like just by editing a text file. And accordingly, your auto grid will take that information and do the changes.
Okay, sir. Okay, sir. Like, so when we write a paper on this docking, we should also mention the configurations also, sir, like in this configuration.
Like in materials and methods.
Maybe like in the results and discussion or some
No,
like.
So all these settings are materials and methods. So what settings you use to do the docking?
Okay.
So you took a grid box of 15 angstrom on each direction with a spacing of this much angstrom, and a grid maps were generated for these atoms. That's how you will write the sentence.
Okay, sir. Okay, sir. Thank you.
40% over.
And sir, can you please show us like how to remove the water molecules and other
Oh, that's pretty easy. You can you can do it manually or you can use PyMol. So let us say I um I randomly take a protein from PDB. Okay, so all these red dots are water molecules. How do I know this? Because oxygen is shown in red by default and it's not bound with any hydrogen. Hydrogens are not there in PDB file by default. So I can know it is water molecule by adding hydrogens. So you can automatically see these are all water molecules, right? So I have two chains here, and there is some kind of a non-protein entity also bound with it. So the easiest way you can do is you can click on the action menu and remove water, and water is removed, but that will remove only water. Now, if I scroll on the sequence, I will see at the end that there are these extra non-protein entities, and then I can just delete them. Now I have only the protein, and I just cross-check it by looking at the sequence here at the top, and I see there is only sequences, amino acids there. So crystal waters, that's what I'm talking about. Crystal waters are not necessarily biologically meaningful. In a biological setup, in a reality, in sorry, in in the cell, the protein encounters less water molecules than a crystal. So crystal waters are more in nature. There are more crystal waters surrounded by protein than compared to the actual number of waters that the protein will actually encounter inside the cell. The other way you can do it is, let us say, I open the PDB file. We So this is the PDB file that I'm opening in a text editor. I keep only the atom records because the water molecule will be in the heteroatom records. So you can see that at the end, up to here, I only have atom records. All these are something that it is used for doing some other calculation. In a particular protein like the one that I showed you in PyMol, let me open in the RCSB website itself. You will see the heteroatom records, het atom ATM, that will start at each line. Water molecule is labeled as HOH. That's the convention. So you can do a grep in the Linux command, take only the atom records, or manually delete them. More than one ways of doing the same thing. So it's not necessary that one is better than the other. You want to remove the water molecules. That's the idea. Let's see what's the status now. 70%. Another 30% more. So another 4 minutes remaining. Any other questions?
So if we wanted to do a dynamic simulation of this conformational change, do we need this molecular docking file, the results file, log file? Like
Uh, do you
the
want to
result of
do do you
the
want to
result
do the
of
conformational study of just the protein or with the drug bound to the protein?
the drug, when the drug comes and binds to the protein, and then after binding, after binding, the change is the protein that undergoes. Like
So then you have to take the complex. So you have to take the docked complex
hmm,
and then you have to use that file, not this output file. This is the grid output file. Whatever you're seeing on the screen right now is the grid output file, GLG, grid log.
okay,
You will get a doc log, DLG, from the DPF file. That DLG file is what you have to use. And in the DLG file, you have to extract the coordinates. And once you extract the coordinates, that is when you can do the simulation of the complex.
okay. So in future, in this course, we are we going to do the simulation, sir?
I hope so. I think there is a module for is Puran around here. Puran is not here, but I know that the previous version of this course we had molecular dynamics simulation and a demo of the molecular dynamics simulation, and um, we showed them how to do a MD simulation and all that. So you will be learning it
Okay, sir.
90%
Thank you,
because
sir.
the syllabus and the schedule topics have changed a bit, but then overall it's still the same. So you will be learning it. Maybe in the more because this is a 6 months course, right?
Yes, sir.
Yeah. So I think as the months progress, you will go to most advanced topics, and there you will have molecular docking simulations and molecular dynamics simulations as well. Okay, last 10%, last 1 minute left. So you can see that even for protein, it's not a big protein. Okay, whatever protein I took is not a big protein. The protein is basically 410 residues in length. Not a big protein at all. And that if I do a blind docking in a normal laptop like what you guys would have in a Dell Inspiron laptop. Sweetie, plug in. My student's name is Sweetie. Her official name is Sweetie. Okay. So don't mistake me if you think I'm calling her by a nickname or something. So we had to her laptop battery is draining and I will not be able to finish the demo. So that's why I had to call her immediately with her name. Okay, so as you can see, the grid parameter file ran successfully. You should be seeing these two words at the end of it, successful completion. Until this grid log file is completed and these lines are written, You remember in the grid, when I launched Autodoc, there was a white box or a window that popped up. That will remain on your screen until the job is finished. If that comes and vanishes immediately, that means there is some missing. So now that we have the macromolecule process, the grid made, everything, the maps have been made. You can look at the maps. The map files are like they will be a little bit larger in size. And these are also again uh, uh, again flat files. And this is how it will look like, mostly zeros all the time wherever there was no hydrogens that it meet. But then in some places it will have values and it will be a huge list of files. It goes on and on and on like this. You will see some values intermittently. That's the map file. Okay. Okay. So let me close this. Let me close this. Let me close this. Okay. So this is the input file. We have the input file ready, which we probably made it with Babel for the AMP. And now I'm going to use this menu to set up the docking file. So first I need to select the rigid file. And since we are doing the easiest one for this demo and I have another 50 more minutes, so I'm going to set the rigid file and the receptor file. I'll select the PDBQT and then ligand. I don't have it on the session here, so I'll open it and it will load. You can see the file here. These are the settings. I'll accept it. And then search parameter. Here's where I can choose from the three: local search, simulated annealing, or genetic algorithm. We use the Lamarckian genetic algorithm, and right now we are going to do 10 runs. Okay. And then I accept it. And then docking parameters you can customize, but most of the time, for most of the things, the default settings are okay. But if you are not getting the results as you are supposed to, then you have to change those and then customize it based on the system that you're using. So don't use the tool as a black box because it will throw out a lot of false positives. And then I'm going to select Lamarckian here as the output, and I'm going to write 396.10923.dpf. So that indicates what receptor, what ligand I'm docking, and that will be the output file name as well. So now my DPF file is made. I can have a look at the DPF file here. So it will tell me what is the small molecule, what is its coordinates, the center of it, what all maps it needs for this molecule, and all these parameters that start with the letter GA. Those are all the genetic algorithm parameters that you can play around with. And we are going to do 10 dockings of the same molecule. So we are docking it 10 times, and that is indicated at the GA run. So now we will start Autodoc. So again, I have to show the path where the actual executable is. It is in program files, Rips Research, Autodoc 2.6, and I have to select Autodoc 4. And I have to select the DPF file. The DLG file is automatically filled, and I will launch. Now this window, it didn't vanish immediately. That means it's running. You will see a DLG file created. So you can open it and it will show all the parameters that we have set up so far. And then it will say beginning genetic algorithm run 1 of 10. Okay. And then it will go 2 of 10, 3 of 10, 4 of 10, and it will run for a period of time. Now there are 27 torsions. I have 15 minutes left. I'm gonna let it run. Okay. But I'm gonna show you the output. So let us say this is the file that the DLG file, it ran. At the end, you will see successful. Again, you should see the words successful completion. That means it ran proper. It will tell you how much time it took. This took 51 minutes. Now, I'll show you each run how it does. So where we saw right here. There it was beginning genetic algorithm docking 1 of 10. So this is the first docking. This is the estimated free energy of binding. This is the final intermolecular energy, final total energy, torsional energy. This 1 plus 2 plus 3 minus 4 is what you get here. Then it will tell you what are the torsions, and these are the coordinates of the docked conformation. Now you can see that here it says docked. So that means this particular conformation, this is the output for that particular docking. We did 10. Now, wherever you see this dock, you will see 10 confirmations. Meaning how many ever dockings you did, you get that many confirmations. But the interesting one that you are supposed to look at is you have to look for the word clustering. Now, clustering is where it will give you a histogram. So this histogram is made up of hash symbols. Every confirmation. Okay, someone is in unmute. Can you mute yourself, Gayatri Chakrathi? Okay, I'll mute you. Okay. So the confirmation, each of the 10 confirmations are pretty different here, and they end up having different clusters. Meaning, if two confirmations are very similar, they will form one cluster. And when they form one cluster, you will see a hash symbol next to it like this. That means that cluster has two entities which form a interaction or same interaction. Now, what is its rank based on? Rank based on the binding energy. Here, everything is positive because it's a peptide. Peptides give positive binding energies in orthodox. That doesn't mean peptide will bind with a higher and negative binding energy. It is just that that the scoring function in Autodog is not optimally written for peptides. It's optimally written for small molecules. So you have to change that or you have to rescore them once you have the results that you have obtained. Okay, so that is the understanding of this. At the end, you have the models. So you can see run 3 is the highest ranked confirmation of this. Now, let us quickly go and see how much run, whether the first run got over. No, the first run did not over. Okay, so it's going to run for a lot more time than we have. So what I'm going to do is I'm going to kill this job. Yes. Yeah, okay. Okay. And then I can load the DLG file with this analyze menu. So I can go to dockings, open, and then in this, let us randomly pick something. Okay, it will load the output file. Okay, so this is the peptide that is shown. Now under analyze, you can go to clusterings and you can show the clustering. Now this is the histogram. You can see that there are more confirmations on this side and one confirmation on this side. So there is more on that side. And now I can play the confirmations by energy. So the starting one, what you saw is completely the input file confirmation. That is not the docked confirmation. Since this is the molecule and you will not be able to see it properly, I'm going to make it like this. Okay, there you go. Okay, so that is the first confirmation. That's where it docked. Then I can go to the second rank. It docked here. Third rank was close by. Fourth is back to where the first one was. Fifth is back to here. Sixth is here. Seventh is here. Eighth is here. Ninth is here. 10th is here. Okay, so I can play around, play all the confirmations, and one by one, I'll see that when I did a blind docking, that there are two places on this protein where the peptide was more happy to bind. Okay, if let us say instead of 10, I did 100 dockings, then I'll have a good statistics where 50% I will see on one side, 50% on another side. Okay, so what do you do now? You from this DPF file, the DLG file, you have to take this atom records, which starts with the atom. Remember earlier place I showed you docked? Yes, Shiva Bhargavi, do you have a question?
No, sir, no.
You tagged Radhakrishnan ji, so that's why I'm asking. He's
Sorry,
off.
mistake. It's for mistake.
Okay, so you have to copy this atom records, or this is the confirmation of the docked peptide. Then you can combine this with your protein peptidb file, and then that is the dock complex. Now that you can take it for the next step of either rescoring it or for the MD simulation or whatever you want to do. Okay, so that this is the output. This atom records in the DLG file after the clustering records. Below the clustering records, the RMSD will be there and a little bit more outputs. And then you have the lowest energy docked confirmation from each cluster. That is what is the output that you need to use it for the like MD simulation, molecular dynamic simulation, or something else. Okay. So with this, I'm going to stop and I can take questions.